Imagine holding the power to rewrite the very code of life itself, a tool that could alter our DNA, remove disease, and even change the path of human evolution. It sounds like science fiction, but in today's world, this technology already exists. That technology is called CRISPR, short for clustered, regularly interspaced, short palindromic repeats. A strange name for something so revolutionary, yet its discovery has opened the doors to possibilities we never thought we'd see in our lifetime. To understand the impact of CRISPR, we must first step back and look at the language of life itself: DNA. Every living thing on Earth carries DNA, a blueprint written in 4 chemical letters. This code determines everything from the color of your eyes to how your body fights disease. For centuries, humans could only observe and wonder about this code. We could map it, study it, and slowly begin to understand it. But to actually edit DNA with precision remained an impossible dream until CRISPR entered the scene. CRISPR was not invented by humans in a laboratory. Instead, it was discovered in nature as part of a bacterial defense system. Bacteria use CRISPR to recognize and destroy invading viruses, much like a microscopic immune system. Scientists realized that if bacteria could use CRISPR to cut viral DNA, perhaps humans could adapt this system to edit genes in any living organism. That realization sparked one of the greatest revolutions in modern science. CRISPR works like a pair of molecular scissors. Guided by a piece of RNA, it searches for a specific sequence of DNA and cuts it at precisely the right spot. Once cut, scientists can then remove, add, or rewrite the genetic code. What once took years of painstaking trial and error in genetics can now be done in a matter of weeks, sometimes even days. CRISPR has turned gene editing into something fast, affordable, and astonishingly accurate. The first and most obvious application of CRISPR is medicine. Imagine a world where genetic disorders like sickle cell anemia, cystic fibrosis, or Huntington's disease can be erased before they ever manifest. Clinical trials are already underway. In some cases, CRISPR has shown promise in correcting genetic mutations in real patients. For families burdened by inherited diseases, this is nothing short of miraculous. But medicine is only the beginning. CRISPR could also reshape agriculture, creating crops that resist drought, pests, and disease. It could engineer livestock to be healthier, more productive, and less prone to illness. And then comes the most controversial possibility: using CRISPR not just to cure, but to enhance. If we can remove disease from embryos, could we also design babies with higher intelligence, greater strength, or specific physical traits? This idea, often called designer babies, has sparked intense ethical debates. On one hand, the potential to eliminate suffering is extraordinary. On the other, the risks of misuse are enormous. What if only the wealthy could afford genetic enhancements, creating a society where some humans are biologically superior to others? The echoes of past eugenics movements send a chilling warning. And yet, history shows us that once a technology is possible, it rarely remains unused. The question may not be whether humans will use CRISPR to enhance themselves, but when and how. CRISPR also challenges our ideas about evolution itself. For billions of years, natural selection shaped life through slow, random mutations. Now, for the first time, humans have the power to direct their own evolution deliberately. Think about that. We are the only species on Earth that can consciously decide to change its biology. Evolution, which once took millennia, could be guided in mere generations. Some scientists even suggest that CRISPR might extend human lifespans by repairing the genetic damage that accumulates with age. Could we one day use gene editing to delay or even reverse aging itself? Others speculate about enhancing the human body for space exploration. By editing genes to resist radiation or adapt to low gravity, CRISPR could help humans survive on Mars or beyond. But with such power comes equally great danger. Editing DNA can have unintended consequences. A small error in the code could trigger harmful mutations or even create entirely new diseases. One experiment in China shocked the world when scientists announced the birth of twin girls whose genes had been edited with CRISPR. The global scientific community condemned the act, warning that the long-term effects were unknown. This event made it clear: humanity stands at a crossroads. The temptation to push the boundaries of CRISPR is immense, but so too are the responsibilities that come with it. Governments and international organizations are now racing to establish ethical guidelines. The question remains: how do we balance innovation with caution? Beyond human health, CRISPR could reshape entire ecosystems. Scientists are exploring gene drives where edited traits spread rapidly through a population. This could, for example, wipe out malaria-carrying mosquitoes. Yet altering an entire species carries unknown risks. Removing one element from an ecosystem can trigger cascading effects, potentially disrupting delicate balances in nature. Some researchers see CRISPR as a key to biodiversity preservation. Imagine bringing back extinct species or helping endangered animals adapt to climate change by tweaking their genes. This idea, known as de-extinction, suggests that one day we could revive species like the woolly mammoth, not from cloning, but from editing modern relatives. But again, the ethical dilemmas loom. Should humans really play the role of nature's architect? Do we have the wisdom to guide evolution responsibly? Critics argue that we should first use CRISPR to fix problems humans themselves have caused: pollution, disease spread, and food insecurity. Enhancements and experiments should come later, if at all. Supporters counter that delaying too long could mean losing opportunities to save lives or solve global crises. Progress, they argue, must move forward. As the debate rages, one truth remains clear: CRISPR has already changed science forever. The genie is out of the bottle, and there is no going back. Just as electricity, nuclear power, and the internet transformed society, gene editing will shape the 21st century in ways we cannot fully predict. Imagine a future where hospitals cure diseases by rewriting DNA rather than prescribing drugs, where parents no longer fear passing on genetic conditions to their children. Or imagine a darker path where inequality deepens, and nations compete to create genetically enhanced superhumans. The story of CRISPR is a story of possibility, promise, and peril. It is a tool unlike any before,